Okay, so it's 9 o'clock. Let's get started. Hello everyone. Welcome to our today's Museum seminar. I'm really excited to welcome our today's speaker, Dr. Thomas Korn from Germany. So Thomas currently is a professor and also the director for the Institute of for Experimental Neuroimmunology in Technical University of Munich School of Medicine and Health. So I knew Thomas quite well because although we didn't do postdoc together, but we actually came from the same lab, did the postdoc. So Thomas actually completed his MD training in medical school, University of Würzburg in Germany, and also same time the Royal London University of Medicine. And afterwards, he did almost a 6 years residency in clinical neurology. And then same time, moved to, he started his postdoc training in neuroimmunology and then become a junior group leader in Department of Neurology, University of Hamburg. And then from 2005, Thomas actually moved to US, actually joined the Harvard Medical School as a postdoc research fellow, started his basic research training. And then since 2008, Thomas moved back to Germany as a senior physician in neurology. And it's almost the same time he started his independent research program, also in the Technical University in Munich, and all the way up to a full professor of neurology. And then, as I just mentioned, since 2017, he became deputy chair of the Department of Neurology. And then 2019, he was further promoted as a director for the Institute for Experimental Neuroimmunology in Technical University of Munich. So Thomas has a long standing interest in studying immune cells in the central nervous system. As we know that uncontrolled adaptive immune response in the CNS can cause a lot of different disorders, which including the multiple sclerosis, I guess. I think this is also one of the main interests for Thomas' group. His group is mainly interested in characterizing the properties of T cells, which induce tissue damage in the CNS, and also to understand how they are regulated. To this end, his group is mainly studying four major areas of research, which including the cytokines, the Treg cells, immune cell trafficking, and also disease monitoring. So Thomas really actually did a stellar postdoc training, also a very outstanding discovery after he became independent. Based on these achievements, he got numerous awards, which including the Hans Webert Award for Clinical Neuroscience, ERC Consolidate grant, and then just last year he also awarded as Sobek Award. Last, I just want to mention during both his postdoc and then independent phases, Thomas really make a remarkable discoveries in the CNS immunology. As I just mentioned, we used to work in the same lab as postdoc researcher. Although he had already left, but by the time I started, but I think Thomas absolutely was until the day I left, one of the most successful postdoc. And then he continued his outstanding work throughout his independent career. It's really exciting to have you on the platform, and then we are very much looking forward to your talk, Thomas. Please go ahead.
Yeah, Juan, thank you very much for that introduction. I don't think that I've ever had such a nice introduction to any conference that I was attending, but I am very much looking forward to this. And what I would like to do with you today is actually to discuss a novel function of B cells. And as John mentioned, so we were both working with Vijay on T cells, and this is still our main interest. But more recently, we actually became interested a little bit into B cells, and I show you why in a second. So the disease paradigm that I want to discuss with you today is actually not multiple sclerosis, but neuromyelitis optica. And you may have heard of this. People previously thought that this is a subset of multiple sclerosis because in these patients, it's mainly actually the spinal cord and the optic nerve that is affected by inflammation. And as you can see here, this is actually a patient of ours that we have in our center. The inflammation here in the spinal cord can be pretty traumatic, and usually it's a longitudinally extensive myelitis that is found in these patients, extending over more than 3 segments. And if these people are not treated well and are not treated in time, they will actually accumulate quite a bit of handicap and end up actually wheelchair bound in no time. So this is a pretty severe disease. And what makes it very interesting to us and others is that in contrast to multiple sclerosis in this disease, we actually know the antigen. And I'll come back to that in a second. But before I do this, I want to give you a bit of background on the clinical course. So we have here in the top row, this is the typical course of multiple sclerosis that you have seen probably multiple times. So this is a disease in multiple sclerosis where you have relapses first, and then after some time, usually after 10 to 15 years, maybe half of the patients go into something which is called secondary progressive disease. Yeah? So this is not very well understood what is the biological substrate of this. So why do these people deteriorate even if they don't have any relapses anymore? This is one of the biggest problems in MS research at the time, because we can treat those relapses pretty well, but we cannot treat the secondary progression. So this is as a background. So, but now in neuromyelitis optica, which you see down here, you always have relapses, and if the patients actually stay with some deficit, this is always connected to a relapse. So we very rarely, I mean, you never say never, but it's very rare that these patients actually go into a secondary progressive disease phase, yeah? Which means that if you treat these patients well in the beginning, so if you are able to control the relapses, you basically actually can save them and can prevent them from accumulating handicap, which is basically not the case in multiple sclerosis. So that's a big difference. And that already tells you that the tissue response probably in these 2 types of inflammation must be very different. So that's one of the differences. Other difference, which is very obvious, and clinicians know this, is that the drugs that are used to treat these different conditions are very different. So it's even the case that some of the drugs that we use in multiple sclerosis, like for example S1P modulators or also natalizumab, they make the disease worse in NMO. So you don't use them. Yeah? So you must not use them, these 2 drugs in NMO. What works in both cases is actually B-cell depletion. And we will come back to that. And the third point I want to stress is that in multiple sclerosis, so if you have multiple sclerosis patients, they rarely have other autoimmune diseases. And this is very different for neuromyelitis optica. So they are these patients are very frequently associated, or they have very frequently also other autoimmune diseases. So there is an association in the same patient with other autoimmune diseases, including myasthenia or lupus or Sjögren's syndrome. And this already tells you that maybe something deeper is going wrong in these NMO patients that is not only associated with the target antigen, aquaporin 4. And this is now what I wanted to tell you. Aquaporin 4 is the target antigen in neuromyelitis optica. So this is a water channel, which is expressed in astrocytes. And it's a very complicated protein. So it has 6 transmembrane domains and forms actually tetramers. And in astrocytes, not only tetramers, but also supramolecular structures, which are called orthogonal arrays of particles. And this is a freeze fracture representation of this. So this is particularly the M23 isoform that actually forms these structures. And it is very clear, so that's a lot of research has been done on that actually the main immune pathology is mediated by antibodies that bind to this water channel. And then these antibodies will fix complement, and that will lead to the lysis of the astrocytes, and that will actually cause the damage, basically. And then you, of course, have a lot of secondary problems. I mean, oligodendrocytes can also be actually in this inflammatory response then be affected, and you can also find demyelinated areas actually in NMO, even though the primary target clearly are astrocytes. Okay, and so, however, it's very clear that in humans, this is an antibody-mediated disease, but since we are a T-cell lab and we actually wanted to understand the T-cell response against aquaporin 4, and also for the reason that these antibodies are clearly class-switched antibodies. Yeah? So they are in humans, they are IgG1 most of the time. And it's very clear that they are also hypermutated. So it's pretty obvious that they must have gone through a germinal center reaction. So there must be T-cell help in order to actually generate these antibodies. So they are class-switched. And then also, this is a bit of a debate, but some people think that even though this is primarily driven, the immunopathology by the antibodies, you may also need T cells in order to, make it possible that these antibodies access the astrocytes. So they must somehow go across the blood-brain barrier. So maybe therefore you need T cells. You do find also T cells in the lesions, and you do find cytokines that are associated with T cells in the CSF of these patients. But these are, in my mind, weak arguments. What is the strongest argument that you need a T-cell response is that these antibodies are class-switched and hypermutated. So they are probably germinal center antibodies. So this was basically the starting point of this project. So we wanted to understand the T-cell response against aquaporin 4. And we started with a very classical experiment. So this is a very old school experiment. So first, actually, we generated the full-length mouse aquaporin 4, so which is very difficult. It's a very difficult protein. It took us a long time, but at the end, we finally succeeded. So we had the full-length protein, and we immunized the wild type, just a regular wild type Black 6 mouse with a full-length protein. And then after priming, so after 8 to 10 days, we took the draining lymph nodes and we did a recall response with overlapping peptides that covered basically the whole sequence of the aquaporin 4. And the overlap was by 15 amino acids, so that we did not want to miss any epitope. But this is a classical experiment when you actually want to find out what is the naturally processed epitope of aquaporin 4. And you can already see that none of these epitopes actually gives a convincing response. There may be a little bit here in this area, but basically it's flat. And when you repeat that same experiment and deplete the regulatory T cells, because you can always argue that maybe you have a very strong Treg response, you basically have the same outcome. And that already tells you that it's probably a central tolerance phenotype that we are looking at here. And that is also corroborated by the fact that if you do that same experiment now in a aquaporin 4 knockout mouse, for which actually aquaporin 4 is a foreign antigen, you do get quite a bit of a response, and you see that the peptide 41 is actually the one that is most likely the major immunogenic epitope presented in the mouse MHC class 2 context IA of B, which is the class 2 complex of Black 6 mice, of course. And well, that made us wonder whether we are looking here at mainly at a central tolerance phenotype. And so we wanted to look at this a little bit more into detail, and so we needed tools. And the major tool that we generated based on these data was actually a tetramer. And we used here the Mark Jenkins protocol to make tetramers. That actually is peptide 41, which is aquaporin 4 205 to 215 in the context of IA of B, so that we could directly measure these T-cell responses. And so first question we had is, what is actually the relevant antigen-presenting cell that presents aquaporin 4 in the thymus? Because if it is centrally tolerized, the thymus is most likely the organ where this tolerance is induced. And we started with a very simple experiment because, as you know, for example, dendritic cells can actually present autoantigens in the thymus. But of course, the major antigen presenters in terms of autoantigens in the thymus are medullary thymic epithelial cells. So these are stromal cells in the thymus that are radioresistant. And this is the basis for this experiment. So here we put basically bone marrow of a donor mouse into a irradiated wild-type mouse. Yeah? And we did this in a crisscross design with aquaporin 4 knockout or wild-type mice. And that way we can actually look at aquaporin 4 sufficient or deficient stromal cells and aquaporin 4 sufficient or deficient hematopoietic cells, and then read out the T cell response against aquaporin 4, and we use the tetramer to visualize this. So let's look at the controls first. So this is basically a reconstruction of the global knockout mouse, and you can already see here on the top row is a control tetramer, just to give you an idea that the background is actually very slow, very low. And here you can see a clear response. So this is basically a knockout mouse for which aquaporin 4 is a foreign antigen, so you see a clear response. This is the control wild-type mouse, so wild-type bone marrow into wild-type recipient, and you don't see basically anything in terms of a T cell response. And so this is the condition where we would have expected something, because here we put wild-type bone marrow into aquaporin 4 knockout mouse, and this means that in this mouse, the MTECs, so the medullary thymic epithelial cells, are knockout for aquaporin 4. So they would not be able to present this to the nascent thymocyte repertoire, but we did not see anything in this condition. That was very surprising. But we saw actually something when we put aquaporin 4 knockout bone marrow into wild-type mouse. So here, the hematopoietic cells are knockout, but the thymic epithelial cells are sufficient in aquaporin 4, and we still do see a bit of a response. So if you do multiple experiments, this is clearly more than in the wild-type to wild-type situation. It's maybe not as high in terms of a response as in the global knockout, but still, it's a clearly above background. And that was interesting because that pointed out that maybe hematopoietic cells present their endogenous aquaporin 4 to thymocytes in order to tolerize them. So this is not a totally new idea. So that's been known before. So as I said, dendritic cells can do this. For example, they ask the dendritic cells in the thymus, but they mostly take up the aquaporin 4 from the environment and then, process it and present it. And that we would not have captured with that particular experimental setup because we had a genetic knockout. So that must have been endogenous aquaporin 4, which was presented by any kind of hematopoietic cell. And so if you go down this list, you end up actually up here at the B cells. And so the thymus people, they know that, so I didn't know it actually, or I was not really aware of it, but the thymus has actually some B cells, and people have seen them and try to understand what they do. And there are some, papers around to suggest that they have something to do with the shaping of the T cell repertoire. But that has mainly been done with artificial antigens that people have expressed in them and then found that T cells that recognize these artificial antigens are modulated by these B cells. But now we are looking at a real autoantigen. Of course, the question was, are B cells here the negative selectors of the T cells? And this is just to show you that these B cells sit in quite a strategic position. So they are at the corticomedullary border here, as you can see here. And actually, when we sort them out and look at them in terms of Tachmann here, very simple expression for aquaporin 4, they do express aquaporin 4. So the thymic B cells have aquaporin 4. The thymic epithelial cells, however, also express it. And of course, astrocytes are a positive control, and you can see this is orders of magnitude lower the expression in the thymic B cells as compared to astrocytes, but still it's measurable and they do express it. And the interesting thing is that this is not only true in mouse, so this is a mouse thymus, this is also true in human thymus. So we got actually hold of a neonatal thymus, And we did a B cell staining. We were surprised that quite a few B cells are also present in human thymus here. And some of those, it's not an absolutely rare event. I mean, it's quite, you can find them. Some of those B cells actually express aquaporin 4 also in human thymus, as you can see here. So this is an example. So CD20 identifies the cell here green as a B cell, and the red aquaporin 4 is here in the membrane of this B cell. So that is something that also can be seen in humans. So, but the question is this functionally relevant? And for that, we went back to our mouse system, and we actually did conditional knockouts. Yeah? And since we found aquaporin 4 in thymic epithelial cells and in B cells, we knocked it out both in thymic epithelial cells using the FOXN1 CRE and in B cells using the MB1 CRE with a FLOXed allele of aquaporin 4. And then we basically repeated the same experiment that I've shown you before. So first of all, we looked at the naive repertoire, so without immunization, just naive repertoire of adult mice with these genotypes using the tetramer. And of course, here you only find very few cells, but you can already have the idea that there's almost nothing in the delta text. So when the thymic epithelial cells do not express aquaporin 4, that doesn't make a lot of difference as compared to the wild type situation. But in the B cell conditional knockouts, you do get a bit of a signal here, which is might actually come close to the global knockout situation. And of course, this is enhanced if you immunize the mice and then look at the immunized repertoire. So here you can clearly see that the delta B mouse, in which the B cells are knockout for aquaporin 4, looks pretty much similar to the full knockout, while the delta tech mouse, so the thymic epithelial conditional aquaporin 4 knockout, looks more like the wild type mouse. Okay, and that already tells you that B cells might somehow be involved in the shaping of the T cell repertoire against aquaporin 4. And now, of course, I mean, we did a lot of analysis of these T cells that now can be rescued when you knock out aquaporin 4 in B cells. And I don't want to go into too much detail here, but one interesting thing was that if you then look at the phenotype of these T cells after immunization of the antigen-specific T cells that come back, and that's basically here in this gray box, then you can actually see that some of them, quite a few actually of them, express BCL6, which is a transcription factor that has been associated with T follicular helper cells. And this is after immunization, of course. And that comes pretty close, actually, or maybe, yeah, is in the same range as the fraction of BCL6 cells that you find in the global knockout. And that's also the case for raw gamma T. So basically, the phenotype of the antigen-specific T cells, and this is here gated on the tetramer positive T cells, is pretty similar to the global knockout situation, suggesting that these T cells that are rescued, presumably from negative selection, can go into a TFH-like phenotype in these B cell conditional aquaporin 4 knockout mice. So question now is, since I showed you text, so the thymic epithelial cells also express aquaporin 4. So the question is, are B cells not only necessary, but also sufficient for this, let's call it, negative selection of aquaporin 4 specific T cells? And for this, we created a mouse, a little bit of a complicated system here, where, I mean, you can think of it as a global knockout mouse, where only the B cells are sufficient for aquaporin 4, okay? So, and the control of course, would be a global knockout mouse where everyone, including the B cells, is knockout. So, and when you have such a mouse, and then look again at the T cell response, let's focus on these two cytograms in the bottom right here. Then you can see that if everyone is knockout, so this is basically a global aquaporin 4 knockout, again, you can see a pretty strong or pretty good or sizable response of aquaporin 4 specific T cells. And in a situation where everyone is knockout, including the techs and everything, except for the B cells, so only the B cells would be here sufficient for aquaporin 4, then this response is gone. So that suggested to us that so the B cells are sufficient to do this. So if everyone else cannot do it, the B cells alone, if they are sufficient in aquaporin 4, they would be able to purge the repertoire of aquaporin 4 specific T cells. Okay, so that the model would be like this. So we had, and that's actually been suggested before. So these B cells in the thymus, they are probably peripheral B cells that go into the thymus maybe early in life, and then they would seed that niche in the thymus and be self-renewing in the thymus. But they actually have to probably, since if you look at cells in the periphery, they do not express aquaporin 4, but that means that in the thymus, they probably need to be licensed in a way. And that has been suggested, and I'll come back to this. They probably need some signal that they get in the thymus for them to be able to express and then also present aquaporin 4 to T cells. And in order to find out what that could be, we did a single cell analysis. So we did just a single cell sequencing of these thymic epithelial cells, thymic B cells, as compared to B cells in the periphery to see how they would be different. And if you look very simple, so I don't go into too much detail here, in the single cell analysis, just at the global transcriptome, you can already see that the thymic B cells here cluster together, and they are different as compared, at least in some aspects, different as compared to splenic or blood B cells, or of course, as compared to bone marrow B cells. And when you then, and we ran a lot of, published data on this to find out how they are different. So the one pathway which really stuck out of this was the CD40 pathway. So obviously a fraction, at least, of these thymic B cells do get a CD40 signal. And of course, thymocytes, and that's been known for a long time, they express CD40 ligand, and they could actually give the CD40 trigger to these B cells. And so this is a clear pathway that was actually one of the top pathways upregulated in thymic B cells. So phenotypically, if you look at thymic B cells, there is a trajectory of these cells. So they come into the thymus as naive. So they would be double positive for IgD and IgM. And then they go into this quadrant. So they lose IgD and are IgM positive. And then finally, they would be double negative. And when you follow this trajectory, then both the CD40 ligand, the CD40 signature becomes bigger, when you follow that trajectory, and also the expression of aquaporin 4 is actually going up when the B cells actually go into this licensing process, and the IgM positive, IgD negatives already, so these guys here, they already express aquaporin 4 in a measurable way here by Tuckman analysis. So if you sort them out and measure this, you can actually see this. So that means that this licensing process probably has, we are pretty sure it needs a CD40 signal, which then leads not only to upregulation of class 2 and CD80, but also it leads it to upregulation of these autoantigens, and aquaporin 4 being one of those. And so this is maybe something we can discuss at the end. So the B cell receptor is irrelevant. Yeah, so any B cell does this. I mean, any B cell upregulates aquaporin 4. So the B cell receptor of this B cell does not have to recognize aquaporin 4. And now, of course, the big question is, okay, it expresses aquaporin 4, but can it also present it really? And for this, we actually did a functional assay. And this functional assay is like this. So first of all, we tried to, because you only get very few B cells out of the thymus, So we wanted to have, in order to test this functionally, we needed more B cells. So we tried to simulate these conditions, this stimulation conditions in peripheral B cells. And we actually gave them an anti-CD40 signal and also an IL-21 signal in addition. And you will see already that this actually mimics a germinal center response for B cells. And we can discuss what that means functionally. But if you do this, you would actually induce aquaporin 4 expression in these B cells. So CD40 alone does it. And if you add IL-21, actually even more, a greater expression of aquaporin 4 is induced in these B cells. And when you then on top of it trigger the B cell receptor, the whole thing goes down again. That is quite interesting. We don't understand yet exactly what it means, but we can actually discuss it later. I think it has a meaning. So that means anti-CD40 and IL-21 alone, they would be the best condition to induce aquaporin 4. So, okay, so they express it. Can they also present it? And for that, we needed a reporter cell. And that, of course, can only be a T cell that has a specific T cell receptor. And for that, we cloned a lot of T cell receptors that are aquaporin 4 specific, based here on tetramer staining. We sorted them out, cloned them, and put them in a hybridoma that has an NFET reporter, so that by turning green, we can actually see whether the T cell receptor is triggered. So this is just an example of a few of those receptors that we cloned. And then we used this reporter, this hybridoma, and put it together with these B cells, okay? And then we just could read out the GFP in order to see whether the T cell receptor gets triggered. And you can see here already that when you use B cells that have been treated with CD40 antibody and IL-21 coming from a wild type mouse, they would actually be able to stimulate the hybridoma, as you can see here, while if you do the same thing with an aquaporin 4 knockout B cell, nothing happens. And when you then add the antigen from the outside, so give the peptide, the immunogenic peptide, then also the wild type as well as the knockout B cells can do it, because the antigen binds to the class 2 from the outside. And if you use a blocking antibody to the MHC class 2's molecule, then this response is gone again. And that indicates already that B cells somehow can actually channel their endogenous aquaporin 4 into the class 2 pathway. And not only, so this would be in vitro stimulated peripheral B cells. And when you do now the same experiment with sorted ex vivo thymic B cells, you see basically the same picture. Yeah? So as you can see here, when you sort out these thymic B cells from a wild type mouse, they can stimulate the hybridoma. When you sort out those same B cells, the same fraction of B cells from the thymus of a knockout mouse, this stimulation goes away. And again, this suggests that really these thymic B cells can present their endogenous aquaporin 4. So now, does this really happen in the thymus? And for that, we made retrogenic mice and looked in the thymus. So basically what we did here, so we made mixed bone marrow chimeras, where one part of the bone marrow was a retrogenic bone marrow, where all the T cells would have an aquaporin 4 specific T cell receptor, and put that together with a bone marrow that either had wild type B cells or that had B cells which were aquaporin 4 deficient, so did not express aquaporin 4. And of course, this bone marrow part that gives the B cells also has a polyclonal T cell response, okay, or has polyclonal T cells. And then we made these mice and let them sit for 6 weeks, and then we just looked at the thymus. So there's no immunization, no nothing, yeah, just looking at the thymus. And what we read out basically is the CD4 single positive compartment. And when you look at this here in the polyclonal part of the bone marrow, so this comes actually from this part of the bone marrow that also gives the B cells, then you don't see a big difference in the fraction or in the size of this polyclonal repertoire. And that's also not so surprising, because the fraction of aquaporin 4 specific T cells in the normal polyclonal repertoire is of course very small. So you would not be able to see differences. But when you now look at the retrogenic part of the bone marrow, where all the T cells have an aquaporin 4 specific T cell receptor, and here again, look at the single positive CD4 compartment, you see that there is quite a big difference depending on whether the B cells in the thymus are wild type or knockout. So if they are wild type, the single positive compartment is pretty small. And when they are knockout, it becomes pretty big, suggesting that really there is something going on like negative selection in this situation. Okay, and then, of course, the question is, okay, as I told you, you need a CD40 signal in order for thymic B cells to be able to do this. And there were some reports previously that thymic B cells also express AIR. AIR is actually the major modulator. It's actually not a DNA binding transcription factor, but it's a modulator of autoantigen expression in thymic epithelial cells. And some of these B cells obviously also express AIR, and particularly those, the orange ones, where we already knew that also they express a lot of aquaporin 4. So the question was, is this whole aquaporin 4 expression AIR dependent? So this is a bit preliminary, but we did that basically that same experiment, but now the B cells were either wild type or AIR deficient, not aquaporin 4, but AIR deficient. And when you do this and now look at the retrogenic part of the bone marrow, which has the aquaporin 4 specific T cells, there's not much of a difference. And that suggests to us that at least for this particular antigen, we would not think that this B cell mediated negative selection in this case is AIR dependent. But that's a bit of still of an open question, and we probably have to look at this a little bit more in detail. Okay, so but now what does this mean, or how can we use this? Because at the end we are in a neurology department. We want to make a model basically also for neuromyelitis optica. So when we have now B cell deficient aquaporin 4 mice, can we actually induce a response in them that also leads to disease? And the first thing we did is actually, and that we can discuss this, that has still some problems. So we used these mice, so the B cell deficient aquaporin 4 knockout mice, and immunized them now with the full length protein. And that should actually give us a pretty good antibody response also. And so first thing we saw is, yes, we can actually, when we do this antigen specific, we see germinal center reactions here in the spleen. So this is BCL6 staining in the global knockout, which is always our positive control because they clearly have germinal center responses against aquaporin 4. We also do see this in B cell conditional aquaporin 4 knockouts. We don't see anything, of course, in B cell knockouts. They don't have B cells, so actually everything is pretty much non-existent in terms of structure. And the wild types don't have a lot of germinal centers because they, as I said, they don't have any T cell precursors. And so you can look at it also in terms of do we see TFH-like cells? And yes, we see them here based on BCL6 and PD1 in the B cell conditional aquaporin 4 knockouts. And we also do see some germinal center B cells, at least in terms of BCL6 and CD95 expression here in the blue bar in the B cell conditional knockouts. But the real question, of course, is what about the antibody response? And here, this is always a little bit, I mean, here, this is a good example. There is an antibody response here in the B cell conditional knockout, and this is a cell-based assay. So we have basically expressed aquaporin 4 in HEC cells or in LN18 cells in this case, which is an astrocyte cell line. And then you can use them as a bait and put on the serum and see whether they, the serum stains these cells. And yes, that happens in the B cell conditional knockouts, but this is a great example here, but usually it's way lower. So if you look at the titers, you do see a bit of a titer in the B cell conditional knockouts, but it's lower as compared, if I don't have the data here, but if you did this in the global knockout, the titer would probably be here up, so much higher. And that's probably something to do with the fact that the B cell repertoire also is tolerized. So if you have a normal wild type B cell repertoire in a normal wild type mouse where aquaporin 4 is expressed, it's not actually only expressed in the astrocytes, but also in the kidney and everywhere else. So it's seen by the immune system. then the B cell repertoire also gets purged over time, and it's very hard to get an antibody response against aquaporin 4 in these mice. But we do see something in the B cell conditional knockout. So that was actually already pretty promising. And this is just as a comparison for MOG. So many people work with MOG peptides. So here the immune system behaves totally differently. And maybe let's look at the wild type situation. So in wild type mice, you cannot elicit any antibody response against aquaporin 4, even if you immunize with the protein. It's very difficult. In MOG, in terms of a MOG response, here immunizing with a MOG protein, you do get a pretty good response in wild type mice. And that is, we think, mainly because the T cell repertoire in wild type mice has quite a few precursor cells that are MOG specific. So this is not centrally tolerized that much as compared to aquaporin 4. And this is why when you immunize, you can actually elicit quite a strong response, both in terms of T cells and in terms of B cells. And that's totally different for aquaporin 4. So aquaporin 4 is centrally tolerized. No response, even if you immunize a wild type mouse. Okay, but what does it mean now in terms of disease? So here we immunize with a peptide, and yes, we can actually, in our B cell conditional knockout mice, we can elicit an encephalitis-like model. The lesions are a little bit located differently as compared to MOG peptide-induced EAE, so they are more in the midline, and the most striking difference is actually in the retina. So the retina, you have a clear infiltrate here in the aquaporin 4 peptide immunized B cell conditional knockout mice, and you never see anything like that when you immunize with the MOG peptide. So this is also B cell conditional aquaporin 4 knockouts. They do get a regular MOG EAE, no difference to the wild type. But they never have any infiltrate in the retina. And that's because aquaporin 4 is expressed in the retina, but MOG is not. So this clearly shows you that there is a difference in the type of the disease. Okay, so that's nice, and we work on this, and we really want to also now create a model where we also have an antibody response, of course, to look at the lesions and how they look like. This is ongoing, but what I wanted to discuss with you is a little bit on a broader scale, because what we are very interested in is actually, and that we have good indications that that is the case. So it's very clear that aquaporin 4 is not the only autoantigen, which is actually centrally tolerized by B cells. And one way to find out let's look at this first. One way to find out which antigens could actually be presented by B cells is this experiment. So what we did is we looked at CD40 knockout mice, and we sorted the B cells of CD40 knockout mice out of the thymus. So these B cells cannot be licensed to present these autoantigens, and compared them with wild type B cells also sorted out of the thymus. And when you do this and look at the differentially expressed genes, actually you come up with a list that is clearly enriched for autoantigens. And many of these autoantigens are actually membrane-bound antigens against which you would have antibody responses. And you can look at this list. So one of them, for example, is here APP, amyloid precursor protein, quite interesting. Or annexin A2, for example, also quite interesting. And there's a lot of them. And this gives us now quite an interesting list of antigens that we could look at in terms of whether or not they are tolerized by thymic B cells. And I mean, here again, you can see that many of these antigens are actually enriched in different organ systems. And one experiment, this was very, very preliminary, but quite interesting, and it may tell you something also in terms of what that means in biological terms, is actually this experiment. So here, what we did is very simple and very dirty in a way. We just took the mature naive T cell repertoire of a wild type mouse, and then we transferred this repertoire into a T cell receptor alpha knockout mouse that has no endogenous T cells, no alpha beta T cells. And then we immunized these mouse just with CFA to stimulate the immune response very broadly. Yeah. And then we took the serum of this mouse like 1 month later and tested it for autoantibodies by putting it on RAG knockout sections, just to see how many, anti-IgG, we were looking for IgG, antibodies are produced that are autoreactive. Yeah. This was our control. And then we did the same thing with T cells that grew up in the absence of B cells. So this is a B cell knockout mouse, MB1-CRE knock-in, no B cells. So we took the naive T cell repertoire, did the same thing, put it in a T cell receptor alpha knockout, immunized it with CFA, took the serum and put it on and tested it for autoantibodies. And what you find is that the autoantibodies in this situation where the IgG, so the switched autoantibodies, are actually generated in the presence of T cells that grew up in the absence of B cells, is much higher as compared to the control situation where you have the T cells that grew up in the presence of T cells. And that suggests that probably the repertoire of T cells that grow up in the absence of B cells is very much autoreactive, or has a much higher autoreactivity than in the control situation. And that brings me to the summary, and there's a lot of other issues that we could discuss, but just to quickly sum up. So I've shown you that B cells actually can contribute to negative selection for this aquaporin 4, so which is a disease-relevant antigen for NMO. We think that this really works through deletion, while the tolerance to MOC is different. I mean, there is central tolerance to MOC as well, because MOC is expressed by MTEX, so by thymic epithelial cells, but this is leaky. Yeah, you still have MOC-specific precursor cells in a normal repertoire, and this is why when you immunize the mice, you can make them sick, and you never make them sick, or it's very hard to make anything sick in wild-type mice when you do this for aquaporin 4. So the B cell receptor is irrelevant for this. So any B cell can do it in the thymus. The role of MTEX in terms of aquaporin 4 expression is not yet entirely clear. So it's clear that they don't have a big role in negative selection, because if they are sufficient for aquaporin 4 and the B cells are knockout for aquaporin 4, the negative selection is basically gone. So they cannot rescue the role of B cells in terms of negative selection, but they may have actually a role for the conversion of T cells into the Treg compartment. We are looking at this at the moment. And the last point I want to make is this broader thing. So we think that these B cells actually present quite a few auto antigens that may be disease relevant, and we are now looking into this, and this may actually be quite interesting also in clinical and translational terms. And one very last point I want to make, that this is a paper I found recently, so that was last year, published in the New England Journal, and that's actually quite interesting, because these are adult patients, so not children, so adult patients that were thymectomized, even though some of them even didn't have a lot of thymus anymore because they were already adult, and they got heart surgery. They needed heart surgery, and in some of them, the thymus was removed, and in others, the thymus was not removed. And then, they were just followed up. And of course, that's in a very, very crude, but what you can see is that the thymectomized cohort somehow did worse than the control cohort. So they all had they were matched for the same conditions, but the surgery was just made in a way that the one group was thymectomized, got rid of the thymus tissue, and the other was not. And there was a difference in the clinical outcome. There was also, it was very clear that the fraction and the absolute number of newborn thymocytes, newborn T cells, based on tracks here, was lower in the thymectomized groups, suggesting that even at this old age in adults, the thymus still has some output of T cells. But very interestingly, the ones that were thymectomized came down with autoimmunity, some of them. at least, suggesting that maybe even in older age, the thymus has some tolerance-inducing function. Of course, this is not connected here to B cells, but yeah, maybe this is what B cells do also in older age. And of course, this has a lot of implication now when you think about CAR T cell therapy, for example, you would deplete these thymic B cells as well, of course. Okay, this is the end. I just want to thank the people. So this project was basically driven by Ali, who's the first author also on the paper, with basically help of everyone in the lab. So I have a very small lab only, so people have to work together. But I want to stress for sure that this is a project that we did together with Ludger Klein, who is a thymus expert, and we probably would not have embarked on this if Ludger had not been there. We worked together very closely. So this was a very nice experience. And yeah, this is our funding sources. And I'm very happy that you listened to me, and I'm happy to take any questions.